So I told you I would be talking about epidemiology and the overlap with environmental health. And so what does that really mean? So I had mentioned that toxicology is one way to consider environmental health, but epidemiology is fundamental to environmental health. Most of our studies in humans are observational in nature. So we have to think about epidemiology, which is the study of the distribution and determinants of health-related states and events in populations. And so you can really drill down with epi, but it is a way of observing populations, a way of determining in environmental health exposures and outcomes in a given population, a defined population. And it's also how we can apply the outcomes that we learn about into the control of health problems. When we combine it, so that's just epi by itself. When we combine epi with environment, so environmental epi, we're definitely thinking about disease states and how they relate to exposures. So disease states and health conditions in population and their association with environmental factors. And I'll tell you that a lot of the times what you'll see in studies is that we're thinking one-to-one. One exposure, one outcome. But the field of environmental epi, the field of environmental health is starting to, or has started to, very much branch out in terms of thinking about multiple exposures, how exposures interact, and then multiple outcomes. It's a work in progress from my perspective to be thinking about how those all interact and how the best epi methods and best statistical methods can be applied in order to determine what the root cause and association is between an exposure and an outcome when we're thinking in multiple terms. In environmental epi, because we're talking about human exposures and we're talking about observational studies, they tend to also be involuntary. So we're tending not to put people into experiments. We're not tending not to have people in situations where they're forced to be exposed to something. But these are just exposures that are around us every day, all day long. And how do we study them? And what are we learning about those exposures from everyday exposures? And as I've mentioned, these are also included in the occupational environment. So whether involuntary or voluntary, or perhaps not even known in the occupational environment. And again, less control in that situation, usually. So some contributions that epidemiology has had to the study of environmental health: population-based studies. So we know that we have better data, we have good data when we're looking at populations and studying the associations between exposures and outcomes. We know that we're looking at observational data when we're dealing with epidemiological research and epidemiological methodology. EPI has provided study design methodology to environmental health studies, and it helps with understanding the descriptive and analytic studies that are involved in environmental health. So here are some special design considerations when we're talking about environmental epidemiology. Cross-sectional studies are very common because we can study a large population. For instance, if we are looking at something like the NHANES, the National Health and Nutrition Examination Survey, that is considered, while it is a cohort in terms of they're following the US population in general over time, when we look at a given year of data, it's a cross-sectional study. What was somebody exposed to, and what is the outcome at that point in time, and not following them over time. Of course, in a cross-sectional study, one of the caveats is always that there's a temporality issue. So we have to think about the fact that we don't necessarily know everything that happened before. We don't necessarily know the other factors that could be colliding or influencing the outcomes, because we're looking at one point in time usually. Molecular epidemiology is a sub-focus or a subfield of epi that is very specific to environmental health. It's actually what I've been trained in, and we are looking at biomarkers in humans. We're thinking about all different kinds of biologic markers. For example, urine, blood, DNA, hair, nails, teeth. There's so much information that can be provided by each of those biomarkers. Some are more invasive than others, and it really depends on the study, what it is that would be the best matrix, perhaps, for getting that information that you want from a human. And also thinking about exposure mixtures, whether, molecular epidemiology is where we would be thinking about our exposure mixtures, but also what are the best matrices for evaluating those mixtures? Could everything come from blood? Could everything come from DNA? It really depends on your research question, and it depends on your study design, how invasive your population will allow you to be. It also depends on what is the best chemical consideration in terms of the laboratory analysis for whether you can use just one biologic marker or not. Environmental toxicology I've referred to also, and this is where we're talking about risk assessment. And this is also where some experimental design comes into play, because this is where you might see animal experiments or cell experiments in order to determine the conditions for dose response. So the dose of an exposure, which is something that we can't necessarily experiment on humans or determine definitively from humans. So it's much easier to do that in a toxicology lab. And then latency is something to consider. It's something to consider in all of epidemiology, but definitely in environmental epidemiology. As I mentioned already, there's temporality issues with cross-sectional designs, but we always have to think about the latency of a design. What is the time between an initial exposure, if we know what that initial exposure is and when it happened, and then that health outcome that we're measuring? When can we actually measure that health outcome? It really depends on the health outcome. It depends on the exposure at hand and what we already know or can deduce about what the outcomes might be from a particular exposure. Latency can range from seconds with an acute exposure and outcome situation. So you know immediately that a radiation burn might happen, or some other form of chemical burn is going to happen immediately, versus decades. So in the instance of something like cancer, where you might have an exposure, but you may not see the effect of that exposure for a long time, for a decade, maybe for a generation. So this is a way to think about a framework of the environmental epidemiology exposure, thinking from soup to nuts in a way. So we have a source of our exposure, and that can either be stationary or mobile, meaning perhaps for stationary, a factory and a smokestack, or for mobile, airplanes or cars, something that is moving and taking its exposure with it as it moves, versus a stationary source that you're living near it, you're working near it or in it, and you kind of have to go to it in order to have that exposure. But it can also be a stationary location, and those exposures, if they're in the air or in the water, those will move, but the original source is stationary in one place. And that relates then to the emissions. Where do the emissions from the source go? Is it in the air? Is it in the water? Does it wind up in our food sources? Is it in products that we are using on our skin, for instance? So, and they may also be related, but thinking just what is that exposure in terms of that emission from the source? And then if there's, in terms of concentration, if there's the amount that we receive, which is also then related to the exposure, and then whether we are thinking about a single source or a single exposure or multiple exposures that may happen concomitantly, because they're happening in terms of emissions together, right? So we have a product that might touch the skin that is going to have more than just one chemical that we're exposed to. Or from that smokestack from a factory, there's going to be multiple exposures from that smokestack, even if they're only making one product, or they're only doing one thing that makes that smoke. We have to think about the distance we are from the exposure. Is it direct? Is it indirect? How long is that exposure? As I mentioned with acute and chronic, perhaps it takes very little time. You don't need to be too close to it, and you will still have an outcome. Perhaps it would take longer time for that exposure to actually have an effect on you. And it really is dependent on what chemical or physical exposure we're talking about. Each one is unique. But something that is very determined in terms of human health, for sure, There's three main routes of exposure. Oral, so ingested usually, somehow getting into the mouth and getting into our digestive tract, and then being able to get throughout the body through the blood. Inhaled, so we're breathing it in, usually through the nose, but you can also think about inhalation of something like a cigarette. Dermal, which would be the skin. So whether we're touching something, whether we're applying it intentionally to our skin, that's going to potentially be a route of exposure. And of course, like anything else, these routes of exposure can be combined at the same time. It just depends, again, on what the exposure of interest in is. And I'll be getting to that too. I'll be giving a few examples. Then there's, in terms of dose, there's a variety of ways to measure dose, but really definitely thinking about internal dosage. How does it get into our body? How much gets into our body after exposure? And then what's the biologically effective dose? After we experience this exposure, how much does it take from that internal dose to have an effect? And then finally, the health effects, whether it's something that will happen acutely after exposure, whether it happens over time, and then we wind up with a chronic outcome, such as asthma, cancer, obesity, diabetes, something that needs to be controlled at a chronic state, or whether it's something acute, such as an infection or a rash or a temporary cough because there's something in the air, your lungs clear it, and then you're okay. And I'd like to just talk about who some at-risk populations are. And this is not an exhaustive list, but definitely we're talking about older adults today, right? We're finally getting to that part. But I have them asterisked for a reason. There's other at-risk populations that I just want to point out. There's pregnant people, children, and adolescents, and I have them asterisked also, and you'll find out why in a moment. Underserved populations and individuals with chronic illness. These are among the at-risk populations that you may see in the literature. There's a focus on ensuring that those populations have environmental justice, have reduced risk of exposure, and that we're considering them in our design of studies. Now, the reason I asterisked the older adults and the pregnant people and children and adolescents is because they actually, of course, that's all dealing with age, and age falls on a spectrum. And there's something called the developmental origins of health and disease theory. It's also, once upon a time, the original person who developed this theory, his last name was Barker, and it's also been referred to as Barker's hypothesis. So it basically says that exposures that we receive early in life, and that can be defined in different ways, whether it's prenatal, so in utero, or whether that is something that happens during the childhood, early childhood, middle childhood, late childhood, adolescence, whether it's multiple exposures, chronic exposure, one-time exposure, that those exposures are going to affect us throughout life. So adult onset health outcomes may be, depending again on the exposures, may be associated with an exposure that happens early in life, and then you carry it through your life. And there's another sort of off-the-cuff type way of referring to it as cradle to grave. So it would be wonderful. They're very expensive if they exist, the few that may exist. A longitudinal study that follows a cohort from birth until death. That is perhaps an ideal in the situation of DOHAD, because you'd be able to see what happens when they're in utero, all the way through childhood, all the way through early adulthood, middle adulthood, and late adulthood, and see what exposures actually do matter in populations. And so a few resources for you in terms of current research. There's child health and development studies that I'm going to be referring to actually a little bit later on. There's the Dutch Hunger Winter Study, which is a famous, well-known study in epidemiology that looked at Dutch children during World War II, there was a famine and the health effects, and they followed those children throughout time. The Framingham Heart Study is another excellent resource to think about for older adult outcomes, and they've started thinking about exposures. And then also the Atomic Bomb Survivors Lifespan Study, which, as I mentioned, radiation is a consideration and how that may have happened early in life, and then what happens as you get older.